specific. So the major objective for me to do this presentation is twofold. One is showcase the capability of a high throughput screening facility here. We will want to best serve the PIs in the University of Iowa by the facility we have here. We can do all the in vitro play-based approach for your research project. And also we have different automation and libraries that I will elaborate later on. Also, we are working as a mini contract research organization for external users. We do have someone interested to use our facility for screening purpose, majorly for the niche project for the drug discovery, especially early drug discovery. And also with this presentation, I want to seek collaborators, especially on image analysis and data analysis, especially in nowadays with more machine learning algorithm and other AI approach. And that's what my major purpose for this one. To do a little bit detail on the capability, we are major do so-called play-based. On the right side, you can see different kind of standard plates or compatible plates. You can use transwell, plate holder, and microfluidics, organelle plates, etc. for the high throughput screening in our facility. We have capability to handle both standard plates and the compatible plate as well. We have automation system from the single Mantis, that's our new add-up, to microlab, to Cell Explorer. We can do quite different level of automation to handle the different plates that make the work a little bit easier. That's our capability. That's my major objective one. The second one is that we want to do image analysis. Because we are doing high imaging based, we can do high content image analysis. You can see we can do this one by in this specific 10 minutes interval to monitoring the mitosis of the cells. And this, for most of the image analysis, people will be straightforward. But if you turn into more, this just one well with 20 different view, and they are moving in every 5 minutes around 3 hours. So that analysis is a hurdle. So that's why we want to find a collaborator to work together to see if we can find a new algorithm for the analysis. The common question we ask is why you want to do high throughput screening. And this is what I want to say. In vitro play-based high throughput approach generate data set that can build up in vitro models to correlate with in vivo results. You notice I repeat the exact same word on the slides because it is common research approach. Jenga showing the common one element at a time. You take one block off, what will happen will use as a functional study, but this will counter-argue by the elephant and the blind story is that you only see only one part of the sophisticated, complicated biological system, especially human clinics. But if you consider doing that one with multiple dots, you can see that you will see more close to reality of the elephant. That's exactly what we can use the in vitro play-based model system to do. They do in vitro model building with systematic, unbiased approach for the cases. They can also generate a prediction and consequently have in vitro, in vivo correlation so that when you do in vivo research, you have a more focused approach rather than directly put in clinical trial, which is super expensive. So this is the resource we have. We have automation. We can discuss later on. If you want to know the details, I can share the slides with you. This is just an overview, give you the resource we have. We have automation. We have detection system, both high throughput as well as high content. We have a screening library. We have small molecule and a little bit biologics as well. I will go to details in the later slides. We do have a certain capability to do information analysis. We usually use the information analysis to direct what we do on the earlier part. So I will basically show the resource we have here. We have small molecule guided RNA cell bank available assays. I will quickly go through them. For the small molecule collection, we are currently have around 190,000 small molecule collection locally. And we also get NCI, National Cancer Center, nature product fractions here as well. That's around 320,000 different fractions here. Here's the individual information. We have earlier, say, FDA approved drug for repurposing purposes, spectrum library for pilot screening, and also structure approach for cancer correlated. And this gives us a different level of screening to fit different purposes of the investigators. So this is a biological. We have a guided RNA library here, but I should point out, people working with CRISPR usually working with pooled library. We are not. We're working with the arrayed, means each will have individual guided RNA. But we do have a kinome RNA cover all the kinome. And you can see that we have the arrayed library here, guided RNA. I also want to point out we are working to collect different kind of PROTAC inhibitor. If you know the words, is basically target protein degradation, so that by using this small molecule, they can specifically degrade a certain target protein with quite good specificity. We don't have the full kinome wide yet. We have 5 now because the technology only 5 to 6 years old. On the other hand, we do have a collection of 320 different kind of kinases inhibitors locally available. You can see on the right side, we already have done something with different assay to detect them as well. And the last part can show the different morphology. We do both high content as well as high throughput. That's the second library I want to mention. For the assay, I want to say we can cover almost all the assay from biochemicals to cell-based. I think we did a lot of different kind of assays. The 3 categories showing here just an indication. And I want to emphasize is that we are not doing one concentration as most might anticipate from screening. We always do dose response. We always do kinetic. And that's why we're also working on the last point is do in vitro, in vivo correlation to make sure the different assay we use for in vitro play-based can be used later on for in vivo. And here is, we have a different 3 category of relatively new assays I can show you. One is on the left side is 3D spheroid. I intentionally show different kind of cases that the top one is cancer spheroid. Middle one is a mixture, co-culture of cancer stem cell-like cases. The bottom one is ex vivo, human-generated, patient-generated spheroid as well. That's our effort to see 2D assay into 3D assay. In the middle part is, in fact, we come from the cellular level to subcellular level. You can see the 2D organelle. The showcase here is the different kind of mitochondrial screening, mitochondrial imaging case. On the left side is that basically what we show here is the 3D mitosis. We can do kinetic rather than the static, either endpoint or single time point. This basically we can follow the live cell for in the current case 3 hours time frame. You can follow the change to observe the mitosis. I don't want to go to details. We can do the discussion. If you want to know the details, I can do a personal introduction to what we have with individual requests you have. there's a elephant in the room nowadays, COVID-19. So when the research ramped down started because of the COVID-19, we're starting to work on to see if we can do something for this one. Within the 2 months frame, we build up resources for testing COVID-19 show up here. We're starting working with virus. We have different proteins. We work with the University of Chicago to do, we did a screening for them because they can generate a protein, NSP15. We work with them to generate different kinds of screening purpose. And we also have trying to get a pseudovirus biosafety 2 coronavirus surrogate, and we are working to get access to SARS-CoV-2. We already have different kind of cells locally. We have biobank, all the cell lines you see here locally, so we can use them for the purpose of our screening because they are the host cells commonly used in this case. If you consider early on we do the 3D spheroid cases, we can also build up that case for the virus investigation as well. We collect different drugs, and as I pointed out early on, we have different drugs available. We have NCI products, and we already get NCI's agreement to allow us to use this collection for COVID-19 research as well. And we get a collection of antibodies from the hybridoma core in the University of Iowa. We're working on it. I'm working with IDT to get a set of antisense oligos from them as well. This is also in process. Since we are mainly do play-based assay, we have different kind of assay available here for the testing, and we can use the library available to investigate them as well. And at the bottom, you will notice that we are preparing these resources. It's not only for the current COVID-19 per se. We can use the facility for later future pandemic preparation. I want to point out I'm working to get the BSL-3 lab access. That will give us capability to do that in a more direct way to do it. And tell you the truth, in March, I didn't anticipate I will do anything related to BSL-3 or to a certain degree with virus at all. But now we have the capability to do that. We are pursuing to really go to in a BSL-3 lab to directly work with the virus that might turn into a future pandemics as well. So this basically showcased the capability, and the common question people will ask, so what? There's so many models out there. We don't have so much time. We don't have money as well. Why you want to do that? And the bottom left, you can see the common death valley as showcased from bench work to the clinical usage. There are two death valleys show up there. The basic concerns in vitro, in vivo correlation. There's no validated solution to overcome that yet, but my point I want to point out is that by doing in vitro screening, give you a case to narrow down the potential in vivo case that will be later on. But still, I want to point out on the right side, in vitro relevance, the relevance with clinical application is most critical. That smart design for your in vitro assay will benefit for you later on in vivo application. Repeat myself here, the take-home message is that by doing in vitro assay, you can do not only single knockout as in Jenga or multiple knockout as a blind elephant. You can do large quantity of repetitive experiments so that you can see the reality of that elephant. I should point out human biology or biomedical system is not that simple. We are not probably just dealing an elephant, but likely a dancing elephant instead. Acknowledgement, we are part of the Cancer Center, and acknowledge all the collaborators. Early on, I work with Johns Hopkins and other collaborators outside as well. Also, thanks my team for doing this one. And that's all. Thank you very much for your attention. I will wait later on for the questions.